Sir, we are live now, sir. Request your permission to start. Sir,
Hmm.
we are
Then,
live now. I
ah,
request your permission to start with the session. Can I
okay.
say?
Ah, ಕೇಳ್ತಂತು
Yes, sir.
ಸ್ಟಾರ್ಟ್
Thank you, sir.
ಮಾಡ್ತೇನೆ.
Yes, sir. Hari Om, everyone. A warm welcome to the 10th session of Higher Anatomy, a lecture series for the first year BMS students. The exclusive sessions will be taken up by the eminent speakers of anatomy across the country for a period of 27 days, which started from 23rd of June and will be running up to the 17th of July. Before starting the session, let me introduce you to Jignasa. Jignasa is a pan-India platform of Ayush fraternity led by the student community of Bharata and was initiated by ABVP in the year 1998. Its motto is Learn Ayush to Practice Ayush. It involves empowering Ayush students from campus to community. It bridges the gap between Ayush students and policymakers as well as administrative authorities, connecting every aspect of Ayush, including students, academicians, practitioners, scientists, industrialists, and hospitals, all under one umbrella. Think globally, act locally. In last 2 decades, our efforts were to strengthen the Ayush education system and boost confidence among Ayush students. Jignasa initiated various programs in different parts of the country, from grassroots to international platforms through discussions, study circles, seminars, workshops, and conferences. Let me list a few initiatives undertaken by Jignasa. Study circles, lecture series on specific topics, free medical camps, social health status survey, hands-on training workshops, conferences to explore and learn unique aspects of Ayurveda, interaction with eminent Vaidyas and academicians, brainstorming sessions with ICE stalwarts, policymakers, and key stakeholders of ICE community, National Arogya Expo and international conferences, national level seminars, medicinal plant visits, pharmaceutical visits, agitations for the demands of high student community, private internship programs for interested students to learn with acharyas, interaction with traditional healers and visit to explore regional flora, online mock tests, social awareness campaigns on various issues, campaigns for the promotions of various concepts of Ayush system, for social welfare like Swarna Bindu Prashana Camp, Group Surya Namaskara, Ashtashatottari Yoga, and other competitions. Free healthcare assistance to people through local governing bodies during natural calamities and accidents are also provided by the Jignasa organization. In our morning session, more than 500 students joined for the live session and asked their questions regarding the male reproductive system. And so far, for the videos of hair anatomy lecture series, we have received almost 7,000 views on our YouTube channel. Vidya Namanarasya Rupamadikam Prachanaguptam Dhanam. We have seen the inability of common student to afford huge amounts on commercial study platforms, despite their jignasa, that is their burning desire to learn. Jignasa is here to give a helping hand. Learn anatomy with experts, and all we need is your immense interest to learn. We hope to reach as many students as possible who can take maximum benefit out of this. Today, we have Dr. Krishnamurthy sir, Professor HOD Department of Sharira Rachana, SGM College of Ayurveda Udupi. Sir has done his UG in Ayurveda Mahavidyalaya Hubli and PG from SGM College of Ayurveda Udupi, where he is also working currently. Sir has worked as assistant professor for 2 years in SDM College of Ayurveda Hassan, presently working as HOD and professor in Department of Rachira Sharira in SDM College of Ayurveda Udupi. Sir has 16 years of teaching experience and Sir has 16 years of teaching experience. Sir has been a PhD guide and guiding 3 PhD scholars, and he also guided 9 PG scholars in the past. Sir has authored of about 14 articles in national and international journals. Sir has been a resource person in 9 seminars and webinars, organized 4 seminars, and done 8 paper presentation. Sir, we welcome you to the session, and to each and everyone who is watching this, we hope you find it helpful. Sir, now I request you to carry forward with this session.
Thank you, Dr. Karthik, for your introduction. Thank you. Now, today we will discuss regarding the embryology. Embryology is a vast topic. Within 1 hour, 1.5 hour, many topics I will deal with that. Important topics I will deal with it. Now, regarding embryology, it is given in must know. Most of the points are must know. And also 20 marks is allotted for the embryology topic. Therefore, long question may come in embryology. Long questions, most of 10 marks question for possibility is there in embryology. Now, which all 10 marks question will come, I will explain it in detail. Not only that, short answer questions also can come, multiple choice questions also may come. Now, what is embryology? First topic is regarding the definition of embryology and the branches of embryology. Definition of embryology is a science that deals with the process and regulations in the prenatal growth. Means regarding the prenatal development, where the science which deals with the prenatal development of an organism within the womb of the mother is called the embryology. Now it starts from the fusion of the gametes, fertilization till the birth of the newborn. Means till the birth of the newborn, the what all developments occur within the uterus of the mother, that all will come in the embryology. Logy means and study of embryo is called as embryology. Now, branches of embryology, say here the question may come as define embryology and name the branches of it. Or may come for 10 marks. Define embryology and explain its branches. Explain its branches may come for 10 marks. Now the branches when we say it is the general embryology, systemic embryology, comparative embryology, experimental embryology, biochemical and molecular aspects in embryology and teratology. Now what is general embryology? It studies of the development during the pre-embryonic and embryonic period, means first 8 weeks of after fertilization. Usually, the after fertilization of the sperm and ovum, up to 2 weeks, it is called as pre-embryonic period. The developmental period is called as pre-embryonic period. And from the third week to the eighth week, from the third week to the eighth week, it is called as embryonic period. And from ninth week till the ninth month, it is called the fetal period. It is called the fetal period, and the development of the fetus is called, it is called as development of fetus. Now, the embryology deals with the development of the embryo also, as well as the development of the fetus also, and in the pre-embryonic period regarding the formation of the germ layers, bilaminar disc formation that occurs in the pre-embryonic period. Now, regarding the systemic embryology, study of formation of the primordia and their structural and early functional organization into various organs and systems of the body. That systemic embryology is not there for the UG students. It is there only for the PG students. Therefore, nothing to worry about it. But general embryology, basic embryology, regarding the formation of germ layers, formation of the placenta, formation of the umbilical cord, spermatogenesis, oogenesis, that all is there for your syllabus. Now, comparative embryology this deals with the study of embryos in different species of animals. And experimental embryology means understanding the effects of certain drugs, environmental changes that are induced on the growth and development of the embryo and fetuses of the lower animals. That is called as experimental embryology. And biochemical and molecular aspects in embryology deals with the chromosomes, genes, sequencing and regulation. Means the genetics also it's a subdivision, it's a branch of embryology. That is genetics that deals with the genes as well as the chromosomes and the hereditary characters transferring from the parents to the offspring. That all will come in the biochemical and molecular aspects of embryology, nothing but the genetics. Teratology deals with the abnormal embryonic and fetal development. That is usually it may be due to drugs, or it may be due to the birth defects, or may be due to the X-ray exposure to the X-ray or by the pregnant mother, or what all, or it may be the defects during the gametes. That all abnormal embryonic and fetal developments are there. That will come in the teratology. Now, what is embryo? Second syllabus topic is In the NCISM syllabus, it is given that define embryo and fetus. Now, what is embryo? Developing individual during the first 2 months or 8 weeks of intrauterine life is called as embryo. Huh? First 8 weeks of the intrauterine development is called as embryo. Embryo means N means within, and brewing means to swell. To swell within, huh? Within the uterus of the mother, to swell within the uterus of the mother, to grow. Huh? And logos means study of. I told you, the study of embryo is called as embryology. Study of development of an individual before birth is called as embryology. Now, what is fetus? A developing individual from the third month up to the ninth week of intrauterine life, that is called as fetus. Now, in the fetal development, the various organs which are formed will grow in size, whereas in the embryonic period, most of the primordial parts of the organs are formed, and placenta is formed, as well as the umbilical cord is formed in the embryonic period itself. The time period of the formation of embryo is first 2 months after fertilization. First 2 months after fertilization. From the third month onwards up to the ninth month, will be called as fetal development. It will be called as fetal development. Now, regarding the spermatogenesis is the third point in your syllabus. Spermatogenesis and oogenesis. Now, they may come for 5 marks. They may come for 5 marks. Uh, and regarding the spermatogenesis, total 75 days is taken for the spermatogonia. You should explain regarding the formation of the primary spermatocyte, formation of the secondary spermatocyte, and formation of the spermatids by the meiotic division. That should be explained here while dealing with the spermatogenesis. May come for 5 marks. It may come for 5 marks. And spermiogenesis also. Spermiogenesis is a process of conversion of the spermatid into the matured sperm into the matured sperm. The Golgi apparatus forms the acrosomal cap, the nucleus forms the head, the mitochondria forms a sheath of the located in the middle piece, proximal centriole lies in the neck, distal centriole of the cell present in the below the middle piece, and axial filament grows out from the centriole. It is present in the middle piece and tail also. The structure of the sperm. We have asked for the preparatory exam here in our college. For the structure of the sperm is also asked. It may come for 5 marks also. The structure of the sperm may come for 5 marks also. That is in the next. You should write the length of the sperm. It is about 60 micrometer, and 60 micrometer in length, and it has a head, neck, middle piece, principal piece, end piece. The head consists of acrosomal cap. The nucleus is present, which is consisting of haploid number of chromosomes. The neck basal body is present. A mitochondrial sheath is present in the middle piece, which provides the energy. And the axial filaments are present in the principal piece and the tail piece. That all should be explained if it comes for 5 marks. Now we can explain the axial filaments are present in doublets, and as well there is coarse petals are there surrounding it. It has a plasma membrane which surrounds it. That all structure is explained in Indrabir Singh book. That can be explained if it comes for 5 marks. Structure of the sperm may come for 5 marks, or the spermatogenesis may come for 5 marks. Now, hormonal regulations of the spermatogenesis also should be written in the spermatogenesis. Actually, the gonadotropic releasing hormone secreted by the hypothalamus will be acting upon the anterior lobe of the pituitary gland to secrete the FSH and LH. means the follicle stimulating hormone and the luteinizing hormone. The luteinizing hormone stimulates the Leydig cells, and Leydig cells secrete the testosterone hormone, whereas the follicle stimulating hormone is responsible for the development of the spermatogonia. It binds to the protein called as androgen binding protein. It binds to the protein called as androgen binding protein, forms a protein called as androgen binding protein, and the androgen binding protein joins with the testosterone and is essential for spermatogenesis to occur properly. Androgen binding protein joins with the testosterone hormone produced by the Leydig cells and is responsible for the spermatogenesis and the spermiogenesis process to occur properly. These hormonal regulations also you can write it within the short notes. So it is asked for a short answer question. Now, oogenesis also may come for as a short answer question, may come for 5 marks. Huh? Usually, oogonium will be present from birth itself. Actually, third to seventh month in the intrauterine life, primary oocytes are formed, and that primary oocyte from seventh month to the birth, it will grow slightly. DNA replication before birth occurs, and synapses and tetrad formation occurs of the chromosomes, and next it will be in the form of primordial follicle. About 2 lakh primordial follicles are present at the time of birth. At the time of birth, but only about 400 primordial follicles mature. They have given that the primordial follicle resulting in reduction of the primary oocytes in their number to 40,000. First, there will be previously I have told 2 lakhs. 2 lakhs primary primordial follicle will be there, or also called as primary follicle. And this primordial follicle will reduce in number, and they will be at the time of about 40,000 follicles will be there but of degeneration, and among these only 400 mature during the reproductive life of the female. Means the reproductive life of the female starts from the puberty age up to the menopause. Starts from the puberty age up to the menopause, and that the remaining primary oocytes undergo degeneration at different phases of maturation. The division of the primary oocyte is arrested until puberty by the oocyte maturation inhibition factor and produced by the follicular cells surrounding the oocyte. And development after puberty, usually the primary oocyte will be undergoing the first meiotic division, and the first polar body and the secondary oocyte is formed. The polar body is formed just to remove the number of chromosomes. Most of the cytoplasm will be going towards the secondary oocyte. Next, the second meiotic division will occur up to the metaphase stage only. Up to the metaphase stage only, where at the time of metaphase stage of the second meiotic division, the secondary oocyte will be ovulated. And if the fertilization of the ovum occurs, then the second polar body is formed. The second polar body is formed. Otherwise, the second polar body is not formed. It will degenerate. It will degenerate. The secondary oocyte, which is ovulated at the stage of metaphase, will be degenerated. Now, the structure of the ovum at time of ovulation, how it will be, that also you can write it in the oogenesis. Regarding the first polar body will be present, the second polar body is yet to be formed. The second polar body will be formed after the fertilization. After the fertilization means the oocyte which is liberated at the time of ovulation, only one polar body is present, and their cells surrounding the oocyte will be called as corona radiata, and a glycoprotein layer called as zona pellucida is present in between the cells of the corona radiata and the oocyte. There is a perivitaline space is there surrounding the cytoplasm of the secondary oocyte. The secondary oocyte will be at the stage of metaphase stage of cell division. Metaphase stage of cell division. Now, next syllabus topic is regarding fertilization. What should be written in fertilization? First, you should write the definition of fertilization. What is fertilization? Fertilization is the process of fusion of two highly specialized, highly differentiated, mature haploid germ cells, an ovum and a spermatozoon, resulting in the formation of unspecialized, undifferentiated, diploid, mononucleated single cell, the zygote. The gametes are haploid in number, whereas the fertilized zygote is diploid in diploid. Means the diploid number of chromosomes are formed in the fertilized zygote. Now, where it occurs, that should be written in the ampulla part of the uterine tube, and the various stages of the fertilization should be written. That is approximation of the gametes, contact and fusion of the gametes, and effects of fertilization that should be explained in three subheadings. Approximation of the gametes, the spermatozoon transport as well as the ovum transport, when they come toward how they come towards the ampulla part of the uterine tube. That should be explained. Now, the spermatozoa, after the spermiogenesis, when they are formed in the seminiferous tubule, the maturation of the spermatozoa occurs in the epididymis. Epididymis part of the male reproductive. In the morning class, we have completed regarding the male reproductive organs. That in the epididymis part, the maturation of the spermatozoa occurs. That changes occurs in the glycoproteins of the plasma membrane of the sperm head, and sperm acquire some motility that is called as maturation. Next stage is capacitation of the spermatozoa. It occurs in the female genital tract. After the intercourse in the female genital tract, when it enters, there the capacitation occurs. And it is time required is 7 hours. Changes occurs in the surface glycoproteins. There is removal of the glycoproteins and exposing the zona binding proteins occur in the sperm. Why? Because it should bind with the zona pellucida of the ovum. Therefore, exposing the zona binding proteins will occur. Now, regarding the ovum transport, the fimbria of the uterine tube moves over the ovary at the time of ovulation, and the ciliary beads of the fimbria sweeps the ovulatory mass into the infundibulum, and ciliary beads of the uterine epithelium and muscular contraction of the uterine tube are responsible for the transportation of the ovum into the ampulla part of the uterine tube. That should be written. How the approximation of gametes occur. That should be explained. After the approximation of the gamete, then contact and fusion of the gametes. Point is contact and fusion of the gametes. Here you should write regarding the acrosomal reaction. Acrosomal reaction, mainly the three acrosomal enzymes are there: hyaluronidase, protease enzyme acrosin, and acid phosphatase. And what is the function of it? That should be written in brief. They are responsible for the disintegration of the barriers. Actually, the glycoproteins of the sperm head binds to the glycoproteins of the zona pellucida. Acrosin causes the digestion of the zona pellucida. Acrosin is responsible for the digestion of the zona pellucida around the sperm head. And zona reaction will occur after the zona pellucida is altered due to the release of lysosomal enzymes by the plasma membrane of the oocyte. That should be written. After the zona reaction, the plasma membrane of the spermatozoon fuses with the plasma membrane of the oocyte, and the disintegrating peptide released from the sperm head initiates fusion of the plasma membrane, and the vitelline membrane contains intergene peptides. After that, next changes is in the calcium wave in the oocyte cytoplasm. Contact of the sperm with the vitelline membrane of the oocyte triggers calcium wave. Depolarization in the oocyte cytoplasm, which triggers the important events at fertilization, and secondary oocyte resumes second meiotic division. Now, the second meiotic division occurs at the time of the fusion of the sperm and the ovum. and contact of the cortical granules with the plasma membrane and release of lysosomal enzymes from cortical granules produce vitelline block and prevents the polyspermy. Only one sperm can enter the ovum. That is the cortical granules along with the plasma membrane. They release the lysosomal enzymes from cortical granules and produce a vitelline block to block to the cytoplasm. And enter the sperm leads to the metabolic changes within the ovum that facilitates its development into an embryo. Now, effects and results of the fertilization. That is the third point. Third point, I have told it. First point is approximation of the gametes. Second point is contact in between the gametes, means regarding the destruction of the barriers and the enter of the sperm into the ovum. After that, the results of the fertilization should be written. Fertilization itself may come for 5 marks, or it may come as a sub question. It may come as a sub question. Fertilization be prepared for 5 marks. Completion of the second meiotic division of the secondary oocyte will occur, restoration of the deprived number of chromosomes will occur, and determination of the chromosomal sex of the fetus will occur, and initiation of the cleavage will occur, genetic diversity will occur, and embryo contains only the maternal mitochondria. Only maternal mitochondria is present, sperm mitochondria is discarded. Means whatever the mitochondria an individual has is derived from the ovum of the mother, not from the sperm. After that, next syllabus topic is this all is must know topic. This all is must know topic. Whatever I have explained before is all must know. And a 10 marks long question possibility is there, and 5 marks questions are also possibilities are there. Cleavage may come for 5 marks. You should be ready for 5 marks. Cleavage is a process of repeated mitotic division of unicellular zygote within the zona pellucida, giving rise to increasing number of smaller cells called as blastomere. What is the duration should be written? It is lasts for 6 days. And there are subdivisions of the cleavage should be written. First, the stage of compaction will occur, starts at the third division, that is 8 cell stage. Maximum contact between the cells will occur. Outer cells forms the tight junction and exhibit polarity. Inner cells forms the gap junction. Nutrition to the embryo is the stored food in the blastomeres and also the trophoblast also provides the nutrition and are transferred to the zona pellucida. Means the cells of the corona radiata also provides the nutrition. Next, formation of morula. Morula means 16 cell stage. Now, 3 mark question is not there for you people. Previously, morula was asked for 3 marks. Now, cleavage can be asked for 5 marks. But morula itself for 5 marks is a small topic. Morula was asked for 3 marks in the previous exams. What is morula? Morula occurs on the third day after fertilization. It is the 16 cell stage of the embryo. It is the 16 cell stage of the embryo where the embryo zona pellucida will be there as well as the trophoblast will be there. Outer layer of cells will be forming the trophoblast as well as the inner cell will be forming the embryoblast. Next, formation of blastocyst. Formation of blastocyst also can be asked for the 5 marks question. That complete cleavage should be written along with the formation of morula and formation of blastocyst. You can start from the cleavage up to the formation of blastocyst. You can explain. Now, blastocyst, when a cavity appears within the morula, when the cells of the go on getting differentiated up to the 32 cell stage and 64 cell stage, a cavity appears which will be filled with fluid at 32 to 64 cell stage on the fourth and fifth day of the fertilization. Usually, the trophoblast gets differentiated into two types. One is called the mural trophoblast and another one is called the polar trophoblast. Trophoblast is responsible for providing the nutrients to the developing embryo. It will be responsible for the formation of placenta afterwards. It is responsible for the formation of placenta afterwards. And polar trophoblast is the part where the embryonic inner cellular mass is there. That pole is called as embryonic pole, whereas the opposite pole is called as abembryonic pole. Opposite pole is called as abembryonic pole. Now, what is why it is called as blastocyst? It resembles like a cyst, and the cavity of the cyst is called as blastocyst, which is filled with fluid, and the inner cellular mass is differentiated here from the trophoblast, and the trophoblast is divided into mural trophoblast and polar trophoblast, and the inner cellular mass will be responsible for the formation of the embryo proper. Next, implantation. Implantation usually occurs on the from 8th day to 12th day, or some books they have given from the 6th day onwards. From the 6th day onwards, the implantation will start, and that implantation will occur in the uterine endometrium, where the site of implantation will occur near the fundus part of the uterus on the posterior wall. On the posterior wall near the fundus part of the uterus, the implantation will occur. Next, formation of the three germ layers. Now, next syllabus topic is regarding the formation of three germ layers. It is a very important topic. It is a very important topic. May come for 10 marks. Formation of the three germ layers may come for 10 marks. From where to write? From the blastocyst formation, you can start from the blastocyst formation. In the blastocyst, there will be inner cellular mass will be there, and a trophoblast will be there, which is called the mural trophoblast, and the polar trophoblast are present. And from the blastocyst, now the cells of the inner cellular mass gets differentiated, and they become flattened. The inner cells becomes flattened to form a thin layer called as hypoblast. Remaining cells of the inner cellular mass will become columnar, and forms the epiblast, that it can be started from here. It can be started from the blastocyst. It can be started, and regarding the formation of the three germ layers should be written in detail. It is an important question. Again, I repeat, if it is important question, may come for 10 marks. Now, the other cells which are there inwards will become columnar, and it will be called as epiblast. First, bilaminar disc is formed in the pre-embryonic period. In the pre-embryonic period means first 2 weeks. First 2 weeks after fertilization, first 2 weeks is called as pre-embryonic period. After that, the embryonic period ನಲ್ಲಿ formation of the procaudal plate as well as the formation of the primitive state that will be coming here. Now, amniotic cells will be formed from the trophoblast, and amniotic cavity is formed. After the formation of the cavity, the hypoblast will be responsible for the proliferates and forms a membrane called as Hughes membrane, and it will be completing the blastocele. The cavity of the blastocyst will be covered completely, and it will be called as primary oxide. Now, two cavities are formed. One is the primary oxide, and another one is the amniotic cavity located above the epiblast. After that, the cells of the hypoblast near the cranial end becomes columnar, and they will be called as procaudal plate. They will be called as procaudal plate near the head end. Whereas near the tail end, the ectodermal cells proliferate. Next is the ectodermal cells proliferate, and is responsible for the formation of a streak called as primitive streak. Called as primitive streak. Now, according to the new textbooks of embryology, previously for epiblast and hypoblast, they were telling ectoderm and endoderm, but the new edition It is not like that. Huh? That is, they are called as epiblast and hypoblast only. Epiblast and hypoblast only. Epiblast will be responsible for the formation of primitive state. Therefore, those who have old embryology book, please go through this. Old embryology, 8th edition, 3rd edition, third in the old edition books, usually the epiblast is called as ectoderm and the hypoblast is called as endoderm. But it is not like that now. The theory is changed. Now, the primitive stage cells, they will migrate in between the hypoblast and epiblast, and they are responsible for the formation of the endoderm also, a inner layer which pushes the hypoblast, forming the endoderm. Next, the remaining cells of the primitive stage proliferate and forms the mesoderm, and the remaining cells in the epiblast will be called as ectoderm. Now the three germ layers are formed. Like that way they have explained. Means all are derived from the epiblast only. According to the new theory, all are derived from the epiblast only. The epiblast cells near the tail end proliferate to form the primitive streak, and from the primitive streak, the the endoderm cells are formed, as well as the mesodermal cells are formed, as well as the remaining cells will be responsible for the formation of ectoderm. Like that, where the three germ layers are formed, that the process of the formation of three germ layers is called as gastrulation. Means all the three layers are derived from the primitive stick, endoderm, mesoderm, and ectoderm. The remaining cells of the epiblast will be responsible for the formation of ectoderm. Now, derivatives of the three germ layers may be asked for a long question also. It may come as a long question or it may come for 5 marks. What are the derivatives of the ectoderm? How to remember this is those which are located externally and which are communicating with the internal cavities, that will be coming in the derivatives of the ectoderm. For example, the lining epithelia of the skin, the epidermis part of the skin, the mucous membrane of the lips, cheeks, gums, hard palate, as well as the lower part of the anal canal, as well as the mucous membrane of the nasal cavity and the paranasal sinuses, and the terminal part of the male urethra and outer surface of the labia minora and wall of labia majora, and anterior epithelium of cornea, epithelium of conjunctiva, epithelial layers of ciliary body and iris, external acoustic meatus, outer layer of the tympanic membrane, epithelial lining of the membranous labyrinth, that all lining epithelium is formed by the ectoderm. Now the exocrine glands located in the skin, sweat glands, sebaceous gland, mammary gland, they will be derived from the ectoderm only. Parotid gland and lacrimal gland are also derived from ectoderm. Endocrine glands nullae, hypophysis cerebri means the pituitary gland and the pineal gland and the adrenal medulla are derived from ectoderm. And other derivatives of the ectoderm are hairs, nails, enamel of teeth, lens of eye, musculature of iris, ciliary body, part of the vitreous body, as retina, and nervous system. Complete nervous system is derived from the ectoderm. All the neurons and neuroglial cells, remember this, the nervous system and the lining epithelium, main, are derived from the ectoderm, which are the surface lining epithelium, the hairs, nails, the epidermis part of the skin, enamel part of the teeth, outer layers that are derived from the ectoderm. That are derived from the ectoderm and the pia and arachnoid matter of the central nervous system. Why? Because the central nervous system is completely derived from the ectoderm itself, and the surrounding pia matter and arachnoid matter are also derived from the ectoderm only, whereas dura matter is derived from the mesoderm. Dura mater is derived from the mesoderm. Now, derivatives of the endoderm are the inner epithelial lining. Inner epithelial lining means epithelium of the floor of the mouth, soft palate, tongue, tonsil, palatoglossal and palatopharyngeal folds, lingogingeal sulcus, pharynx, esophagus, stomach, small intestine, large intestine, upper part of inner canal. ಇದೆಲ್ಲ ಅಂದರೆ internal GIT. Internal GIT will be mainly the epithelial lining is derived from the endoderm. And not only that, the epithelium of the auditory tube, epithelium of the middle ear, inner layer of the tympanic membrane, epithelium of the respiratory tract, epithelium of the gallbladder and extrahepatic duct system, epithelium of the pancreatic duct, epithelium of the urinary bladder, except trigon, and female urethra, except part of its posterior wall, male urethra, except part of its posterior wall. Posterior wall is derived from the mesoderm, and prostatic part and except part of penile urethra lying in the glans penis, which is derived from the ectoderm. And epithelium of the greater part of the vagina, vestibule, and inner surface of the labia minora that are derived from endoderm. Now, regarding the exocrine glands, liver, pancreas, glands in wall of the gastrointestinal tract, outer glandular zone of the prostate gland, urethral glands, paraurethral glands, submandibular and sublingual salivary glands are derived from endoderm. Endocrine glands mainly thyroid gland, parathyroid gland, thymus gland, and islets of Langerhans. That will be derived from the endoderm. And derived from the mesoderm, all connective tissue, including loose cellular tissue, superficial and deep fascia, ligaments, tendons, aponeurosis, dermis part of the skin, and capsule of the various glands, specialized connective tissue, all the connective tissue, blood, bone, muscles also, muscular tissue also, muscle tissue also, except the musculature of the iris and ciliary body. They are derived from ectoderm. Musculature of the iris and ciliary body are derived from ectoderm. Other all muscles are derived from the mesoderm. Heart, cardiovascular system, spleen, lymphatics, pleura, pericardium, peritoneum, lining mesothelium, dentine part of the teeth, substance of the cornea, sclera, choroid, ciliary body, iris, and part of the vitreous body also, and dura mater and microglia that are derived from the mesoderm. Next, kidney, ureter, trigon of bladder, posterior wall of female urethra, posterior wall of the upper part of prostatic part of urethra, testis, epididymis, ductus deferens, seminal-vesicle, ejaculatory duct, ovary, uterus, means the reproductive system as well as the urinary system. Uh, Reproductive system as well as the urinary system, but urinary system not the complete bladder ಆಗುದಿಲ್ಲ, only the trigon part of the bladder only. Other lining epithelium is derived from the endoderm. Other epithelial lining is derived from the endoderm. The trigon part of the urinary bladder is derived from the mesoderm. And endocrine gland, adrenal cortex, exocrine gland, inner glandular zone of prostate gland. Means the derivatives of the three germ layers may come for 5 marks. Derivatives of the three germ layers may come for 5 marks. Next syllabus topic is loss of heredity in embryology. Heredity means the transmission of the inherited characters from the parents to the offspring is called as heredity. And genes are the structural and functional unit of inheritance stored in the chromosome and are responsible for transmission of characters from the parents to offspring. And genes are arranged in pairs, and at the time of gametogenesis they get separated. After fertilization, new gene pairs are formed, and the position of the genes in chromosome is called as the locus of the genes. Genes may be homozygous genes may be present and heterozygous genes may be present. When two parental genes are identical for a particular character, they are called as homozygous genes. For example, height of an individual is a particular character and both are responsible for tallness, then they are called as homozygous genes. If the character is height of an individual and one gene is responsible for tallness and the is responsible for dwarfness, then it is called as heterozygous genes. Then it is called as heterozygous genes. Like that way, some part about the genes should be written, and next you should explain regarding the Mendel's laws of heredity. Mendel's laws of heredity, mainly four laws are there: law of unit character, law of dominance, law of segregation, and law of independent assortment. Law of unit character means all characters are unique by themselves and now called as genes. They are called as genes. They regulate the expression of these characters during the development of an organism. Law of dominance means if the genes exist in pairs, usually for a particular character, they exist in pairs. But character expressed in the first generation is dominant, and which remains suppressed is the recessive. That is called as law of dominance. Next, law of segregation means two alternative factors for each trait get separated during gametogenesis, and each factor has an equal chance of getting transferred to the offspring. It may be dominant one or it may be recessive one. Next, law of independent assortment. Inheritance of one factor is unaffected by the inheritance of the other. Each gene gets associated independently of the other during its passage from one generation to the other. That is called as law of independent assortment. Next, you can write the combination of the gametes. Homozygous normal, if gets married with the homozygous normal, how the offsprings will be. Homozygous abnormal, if he marries with the homozygous abnormal, how the offsprings will be. Homozygous normal, if he marries with the homozygous abnormal, how the offsprings will be. And heterozygous individuals, if they get married with each other, how the offspring will be. Homozygous normal with heterozygous, how the offspring will be. That with combinations we can write. Means if the abnormal gene is dominant, what will happen? What will happen, and if the abnormal gene is recessive, what will happen is explained. And that in the laws of heredity, you can write it if it comes for 5 marks. Next, intermediate inheritance is there, polygenic inheritance is there, multifactorial inheritance is there, and sex-linked inheritance is there. Means through sex chromosomes, mainly X chromosomes, if the characters are transmitted, then that is called as sex-linked inheritance. Hemophilia, red-green color blindness, Duchenne muscular dystrophy, or vitamin D resistant rickets, etc. Multifactorial inheritance means not character not only follow the polygenic inheritance but also are influenced by the environment also. Means example for stress, stress will lead to peptic ulcer, or heavy work, or sedentary lifestyle. Sedentary lifestyle may lead to diabetes mellitus. Uh, and like that, multifactorial inheritance may occur. And another one is the polygenic inheritance. Characters such as height, weight, intelligence do not follow simple Mendelian law of inheritance. They are controlled by several genes located at different loci having similar effects. Next, intermediate inheritance means these characters do not follow the dominant or recessive kind of inheritance. In such cases, a heterozygote resembles neither a homozygote with both dominant genes nor one with both recessive genes. Example, sickle cell anemia is one condition is there where there is homozygote abnormal, and sickle cell trait is there where there is heterozygote. Next syllabus topic is regarding the sex determination and sex differentiation. Actually, they have asked it nice to know. Therefore, it may not come for the exam. Sex determination and sex differentiation may not come for the exam, but if it is nice to know only. Therefore, 5 marks question it will not be asked. 5 marks question will not be asked. But multiple choice question, nice to know is there. Huh? Nice to know is there. But multiple choice question also, from the sex determination and sex differentiation, they can ask which chromosome is responsible for the determination of a male, X ಅಥವಾ Y chromosome ಅಥವಾ XY ಅಂತ ಹೇಳಿದ್ರೆ, they can make it like this combination of four answers they can do, and they can ask in the multiple choice question. But it is desirable to know Allah, nice to know. Therefore, less questions will be asked in this sex determination and sex differentiation. Sex differentiation usually, in this also multiple choice questions is difficult to form, but the urogenital sinus in the male, what the male derivative will be, the urinary bladder, the or the clitoris or the glans penis, like that way they can ask in the sex differentiation. Actually, the sex differentiation is not explained in detail ಆಗಿ ಬೇಕಂತಿಲ್ಲ, detail ಆಗಿ ಬೇಕಂತಿಲ್ಲ, but may come for multiple choice question only. They are nice to know. They are nice to know. Therefore, sex determination and sex differentiation not necessary to give more stress to it. Next, month-wise development of fetus. If it is asked in the exam, you should write the modern development of the embryo. If Masanumasika Garbhavriddhi is asked, then you should write the Ayurvedic Masanumasika Garbhavriddhi. तत्र प्रथमे मासे कललं जायते द्वितीय शीतोष्णनिलैरपि प्रपञ्चमानानां महाभूतानां सङ्घातो गणः सञ्जायते. That all is there, no? That should be written if it Masanumasika Garbhavriddhi is asked. If the month-wise development of the fetus is asked, they want the modern. They want the modern, and that is available in Tortora. That is available in Tortora. First month fetal development, its length will be about 0.6 centimeter. Head and tail folds are formed. Vertebral column and canal is formed. Four-limb buds appear. CNS is formed. Heart begins to beat. Blood formation occurs in the wall of the oxide. Lens placode and nasal placodes are formed in the first month. Second month, its length will be 3 centimeter. Its weight will be 1 gram. Hind limb buds appear. Digits are well formed. Limbs become distinct. Major blood vessels forms. Eyes move to front. Eyelids are formed. Nasal pits are formed. Ear develops. Skin has a single layer. Endochondral ossification of bones begins. Body systems develop. End of third month, its length will be 7.5 centimeter. Its weight will be 30 grams. Bone marrow is formed. Blood formation begins. Bile formation in liver occurs. Urine starts to form. Coils of intestine enters into the abdominal cavity. Pancreatic islets are formed. Prostate develops. Testes reach iliac fossa. Nose develops as a bridge. And nails develop. Next, end of the fourth month, length will be 18 centimeter. Weight will be 100 grams. Complete skin is formed. Tongue develops. Head is large in proportion to body. Hairs appear in the head. Bones are ossified. Joints begin to form. Face takes human features. Body systems develop. Sex determination can be done. At the end of fourth month, the sex determination can be done. End of the fifth month, length will be 30 centimeter, weight will be 450 grams, lanugo appear, lanugo means body has, brown fat formation will occur, vagina becomes canalized, and end of sixth month, length will be 35 centimeter, weight will be 800 grams, and the blood formation completely by the bone marrow will occur, and skin is wrinkled. Uh, Next, end of the seventh month, our length will be 42 centimeter, weight will be 1350 grams, capable of survival, assumes upside down position, skin is wrinkled, testis reaches inguinal canal. At the end of 8 months, length becomes 45 centimeter, and weight becomes 2,300 grams. That is, length is from the crown means from the head up to the buttock region. That is CR length, it is called as. It is observed in ultrasonography. It is observed in ultrasonography. Subcutaneous fat will appear, skin less wrinkled, testes descend into the scrotum. That occurs in 8th month. Ninth month, length will be 50 centimeter long, weight will be 3.5 kg, and the lanugus shed and nails extend to the tip of the fingers. This is the month-wise development of the fetus. It is explained in Tortora. Tortora, if you go through, in embryology part, they have given the month-wise development of the fetus. Next, fetus circulation, very important topic. Usually, may be asked for 5 marks or may be asked for 10 marks also. Maybe asked for 5 marks or maybe asked for 10 marks. Now, in fetal circulation, what are the special structures located in the fetal circulation? You should, while answering it, you should see that the special structures you have labeled it properly. Means the ductus arteriosus, ductus venosus, umbilical vein, umbilical artery, as well as the foramen ovale. Uh, the fetal circulation is different from the adult circulation. Fetal circulation is different from the adult circulation. First, you should write like that, and you should explain regarding the special structures located in the fetal circulation, and the oxygenated blood enters into the fetus through the umbilical vein, whereas the deoxygenated blood are carried out by the umbilical arteries. There will be only one umbilical vein will be there, whereas two umbilical arteries will be there. Right and left umbilical arteries will be there. That special structures you should the special structures present in the fetal circulation are, first you should label them. Next you should write in detail about the process of the fetal circulation from the umbilical vein to the left branch of the portal vein, from there to the inferior vena cava by the ductus venosus, and from the inferior vena cava to the right atrium, and from the right atrium to the left atrium through the foramen ovale, and from the some of the blood from the right atrium will enter into the right ventricle, and from the right ventricle it will enter into the pulmonary artery, and the pulmonary artery will be connected to the arch of aorta by ductus arteriosus, and that blood will enter into the ductus arteriosus also, from there to the arch of aorta, and from the left atrium the blood which is received from the pulmonary veins. Actually, the pulmonary veins here carries the deoxygenated blood, and the pulmonary arteries will be carrying the oxygenated blood. That is the difference in the fetal circulation. That should be highlighted. In the pulmonary artery it carries moderately oxygenated blood, whereas the pulmonary veins carries the deoxygenated blood. Deoxygenated blood. Why? Because there is no respiration is there. Respiration is not there. Therefore, the pulmonary veins carry the deoxygenated blood. They carry the impure blood from the lungs to the heart. There is no respiration. Is there. Therefore, impure blood only will be entering in. That should be explained in detail, fetal circulation, and the remnants of the fetal circulation should be explained afterwards. Remnants of the fetal circulation means what happens to umbilical artery. Umbilical artery, it forms umbilical vein. Umbilical vein will be forming the ligamentum teres. What happens to the umbilical artery? Medial umbilical ligaments they will be forming. What happens to the foramen ovale? It becomes the fossa ovalis. What happens to the ductus arteriosus? It forms the ligamentum arteriosum. What happens to the ductus venosus? It forms the ligamentum venosum. That remnants of the fetal circulation should be explained at the end. Fetal circulation, be prepared for 10 marks also. It may 10 marks and or 5 marks. 10 marks or 5 marks. It is a very important question which may come for the exam. Next, formation of placenta is asked in the exam. Usually in the university exam, they have asked for 10 marks also. Formation of placenta is asked for 10 marks also. There you should, what is placenta? First you should define it. Placenta is a fetal-maternal organ. It connects the growing embryo and fetus with the wall of the pregnant uterus. It is an organ where there is intimate acquisition or fusion of the fetal organ to the maternal tissue for the purpose of physiological exchange. Means the exchange of the nutrients and the waste products will occur in between the maternal blood and the fetal blood. And it is a circular disc-shaped organ of 500 grams in weight. And what type of implantation occurs in human beings? The interstitial implantation will be there in the human beings. Actually, the deciduum within the uterine endometrium, the fetus gets implanted. The embryo gets implanted within the endometrium part of the uterus. Usually, where the site of the implantation will be, the site of the implantation will be the posterior wall of the uterus near the fundus. Posterior wall of the uterus near the fundus. On sixth day to eighth day, the implantation will occur. And the decidua is the functional after the implantation, the internal endometrium will be called as decidua. And the parts of the decidua should be written. The decidua parietalis, decidua capsularis, and the decidua basalis. And the normal site of implantation should be explained. That I told you now. It is the posterior wall of the fundus part of the uterus. And what is placenta previa? That applied anatomy can be written. And abnormal implantation outside can be written in applied aspects. It may be interstitial tubal implantation or tubal implantation or abdominal implantation may occur or ovarian implantation may occur leading to ectopic pregnancy. Tubal implantation leading to ectopic pregnancy can occur. That should be explained. That is, they have given that syllabus topic and its clinical importance. Formation of placenta and its clinical importance, like that way they are told. Uh, Abnormal sites of implantation also, as well as abnormal forms of placenta is also there. Now, how the placenta is formed, you should explain regarding the three structures mainly responsible for the formation of placenta. That are the trophoblast getting differentiated into syncytial trophoblast and cytotrophoblast, and the extraembryonic mesoderm. And the cytotrophoblast cells will proliferate, and it will be responsible for the formation of primary villi, which will enter into the syncytial trophoblast. And when we take a transverse section of it, we will along the XY axis, we can see the cytotrophoblast in the middle and the syncytotrophoblast surrounding it. There will be lacunar spaces located on the sides of the in the syncytotrophoblast. Lacunar spaces appear. That process you should explain in detail regarding the formation of placenta. It is asked for 10 marks. First starting should be it is formed from the decidua. The three structures which are contributing to the formation of the placenta is decidua as well as the trophoblast as well as the extraembryonic mesoderm. First you start from here. What is decidua? You explain it. Next, the trophoblast cells proliferate and they lost their cell membrane and form the things. I will not go in detail now regarding the process. Why? Because it is a vast topic. Embryology within 1 hour, I can't complete all the topics. But from where to write and where to end, you should write regarding the formation of primary villas, secondary villi, and tertiary villi. That is important. Uh, secondary villi. Within that, there will be extramyonic mesoderm also protrudes in. And when we take a section along the villus, there will be a extramyonic mesoderm in the middle, surrounded by the cytotrophoblast, surrounded by the syn cytotrophoblast outside. Like that way, that secondary villi formation, after that tertiary villi formation also, that should be highlighted. First, differentiation of trophoblast into syncytial trophoblast and cytotrophoblast, formation of primary villi, secondary villi, and tertiary villi, and form after the formation of chorionic villi, uh, the two types of chorionic villi will be there: chorionic frondosome and chorionic leve. Chorionic frondosome means that which is present near the basilis, decidio basilis, that will remain, that will remain, whereas other chorionic villi will disintegrate, and other chorionic villi will disintegrate, and that chorion will be called as chorionic leve. That chorion will be called as chorionic leve, where the chorion will be smooth without any finger-like projection. Villi means finger-like projections will be there. Now, primary villi formation, secondary villi formation, and tertiary villi formation, that should be written. You should explain it stage by stage regarding 10 marks question may come. 10 marks question may come. No more, they have asked it, formation of placenta. I have gone through the old papers. They have asked it for the exam for 10 marks. And cytotrophoblast cells will proliferate and forms a cytotrophoblast shell. After that, structure of the placenta also. In one of the question paper, structure of the placenta is asked. If the structure of the placenta is asked, it is not necessary to write the formation process. Formation process ಬರೆಯುವ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. But when the structure of the placenta is asked, it is discoidal in shape, having two surfaces, maternal surface and fetal surface. It is 500 grams in weight. Maternal surface is irregular and is divided into 15 to 20 small lobules called as maternal cotyledons, and the septa grow into the intervillous space from maternal side, divide the surface into 15 to 20 maternal cotyledons, like that irregular maternal cotyledons that should be explained. And each maternal cotyledons contains of 2 to 4 anchoring villi, and one anchoring villi and its ramifications is constituted one fetal cotyledon. What is the difference between fetal cotyledon and maternal cotyledon? Maternal cotyledon means the part of the placenta located in between the two septas on the maternal surface, whereas fetal cotyledon will be there. There will be 2 to 3 fetal cotyledons within the maternal cotyledon. Fetal cotyledon includes only one anchoring villus. One anchoring villus connected from the fetal side to the maternal side, and its ramifications. The ramus cori, ramular cori, and the floating covilli are located in the intervillous space. Now, regarding the fetus surface, it is smooth and covered with amnion, and umbilical cord is attached to it at now near the center of the surface, and the fetal part is contributed by chorionic frondosome that is seen as a plate called as chorionic plate. From the chorionic plate, 40 to 60 extensions, fetal cotyledons arise and extend towards the deciduum basilis. Where the deciduum basilis, there the maternal surface will be present. Next, classification of placenta. You should, based on shape, discoidal placenta, bidiscoidal placenta, oval placenta, triangular placenta, irregular placenta, lobed placenta, diffused placenta, placenta subcensurata means a small part of placenta is separated from the rest of the placenta. Fenestrated placenta when a hole appears, and circumvallate placenta, where circumvallate placenta means covered in the sides by the deciduous bacillus. You can see in the diagram. Fold of decidua covers the placenta on the margin. That is called as circumvallate placenta. Next, classification of placenta according to the attachment of umbilical cord also. A normal central insertion, a paracentral insertion of the umbilical cord, marginal placenta, or velamentous placenta. A velamentous placenta means umbilical cord is attached to the fetal membrane, means the amnion and the chorion, to the peripheral margin of the placenta. Next, classification of placenta according to the distribution of the umbilical arteries also, dispersed type, magistral type, and furcate type. Like that, the three types are there. Dispersed type means umbilical arteries show dichotomous branching, means progressively from larger lumen to smaller lumen. Magistral type means arteries present uniform caliber up to the periphery of the placenta. So uniform caliber will be there. And that is called as magistral type of placenta. Whereas furcate placenta means blood vessels divide before entering into the placenta. Next, umbilical cord. Umbilical cord also is must know they are given, and formation of umbilical cord may be asked for 5 marks, not more than that. Formation of placenta is asked for 10 marks. Listen, remember this. And but formation of umbilical cord may be asked for 5 marks. Umbilical cord is a tubular in structure and is covered by amniotic membrane. It contains blood vessels, yolk-sac, ligaments, and amniotic connective tissue. One end of it is attached to the anti-abdominal wall of the fetus, and the other end is fixed to the center of the fetus of the placenta. And functions is the umbilical vessels transport oxygen and nutrients from the placenta to the developing fetus and eliminate carbon dioxide from the fetus secretion into the placenta. And regarding the formation of the umbilical cord, that should be explained during the second week of development with the appearance of extraembryonic coelom. The extraembryonic mesoderm is divided into somatopleuric and splanchnopluric layer around the entire conceptus except at one area. This unsplit area of the extraembryonic mesoderm forms the connecting stalk, and the connecting stalk contains the primary mesoderm and suspends the developing bilaminar embryonic disc along with the amniotic and oxide cavities in the blastocyst. Next, with the establishment of cephalocaudal means head end and the tail end of the embryo. The connecting stalk moves towards the caudal end of towards the tail end of the embryo, and an invagination from the tail end of the embryo occurs, known as allantoic diverticulum, into the mesoderm of the connecting stalk. And during the third week of development, extraembryonic blood vessels develop in the chorion and connecting stalk and are known as umbilical vessels. Means what are the contents of the umbilical cord? There will be two umbilical arteries, one umbilical vein, and allantoic diverticulum is there, and water's jelly. And during the fourth week of development, with the formation of head and tail fold and lateral folds of the embryo, the connecting stalk, which is constituent allantoic diverticulum, moves to the ventral surface. From the caudal end, it will move to the ventral surface of the developing embryo, with the incorporation of the yolk sac into the head and tail folds of the embryo, contributing for the foregut, hindgut, respectively. The midgut between the two is in the communication with the extraembryonic part of the yolk sac known as definitive yolk sac. Means there will be a duct is there called as vitalo-intestinal duct. A vitalo-intestinal duct means the connective duct in between the secondary yolk sac is present, called as vitalo-intestinal duct, which will be forwarded towards the umbilicus. The communication between the midgut and the definitive oxygen is that told it vitalo-intestinal duct and lies close to the connecting stalk because of the formation of embryonic folds. The amniotic membrane forms a tubular investment enclosing the connecting stalk along the allantoic diverticulum. Means next, what are the contents of the umbilical cord should be written. Umbilical arteries, two umbilical arteries, one umbilical vein. Actually, the left umbilical vein will remain. First, the two umbilical veins will be there, but one of the umbilical vein will disintegrate, and only the left umbilical vein will remain. Waters jelly, allantoid diverticulum, and vitalointestinal duct will remain. Thank you. In this, I have completed the syllabus. If you have any doubts, you can ask me.
Thank you, sir. Uh, Thank you guys for listening so far, and thank you, sir, for such an elaborative session on such a vast topic, that is embryology. So now we invite you to start posting the questions in our chat box, and sir will be taking the questions from now. Sir, the first question is from Anna, a student
Hmm.
named Anna. Is there a way to
Hmm.
simplify the derivation just so we can remember them easily? That is the first question, sir.
Derivation, no?
Yes, sir.
Uh, See, derivation, there is a simple way is there. How to remember is the lining epithelia which is there on the outer surface uh, is derived from the ectoderm. Hmm? And that part also which communicates from the exterior to the interior means just the connecting part. For example, the mucous membrane of the nasal cavity, the mucous membrane of the paranasal sinuses, the mucous membrane of the oral cavity and the lip, the conjunctiva part of the eye, the terminal part of the urethra. Like this way, the part which is the part which is from connecting the exterior to the interior, that also will be the derivative of the ectoderm. That also will be derivative of the ectoderm. And you can remember that mesoderm is responsible for the formation of cardiovascular system. Mesoderm is responsible for the formation of skeletal, all the connective tissue. Listen here, that is important. All the connective tissue means you can write the bone, you can write the blood, you can write the connective tissue, loose areolar tissue, what all connective tissues are there, that all will come in the mesodermal as the mesodermal derivative. The endodermal derivative when we write, that is the internal lining. Say the internal lining of the yolk sac which is there, no? The yolk sac will be incorporated into the embryo. The yolk sac will be incorporated into the embryo. Therefore, the inner lining of the yolk sac, which is there, which is derived from the hypoderm, that will means hypoblast, sorry, hypoblast. That after that, the endoderm will be lining it. That all internal lining is derived from the endoderm. Means what we can go through the epithelial lining of the gastrointestinal tract. Now the epithelial respiratory tract is also formed from the as a diverticulum from the foregut. Say systemic embryology is not there for you people. Therefore, you people don't know regarding the how the trachea is formed. The trachea as well as the lung alveoli all are derived from the foregut only, from a diverticulum appearing from the foregut. Therefore, what will happen is that the epithelial lining of the respiratory tract, the epithelial lining of the digestive tract, all is derived from endoderm and the epithelial lining of the urinary bladder. How to remember is like this way you remember. Say mesoderm means all connective tissue. Mesoderm, all connective tissue, blood, bone, muscles, muscular tissue also. All muscle tissue except iris and ciliary body. Okay, one. Second point, you remember that the endothelial lining of the interior of the gastrointestinal tract, the interior of the biliary apparatus, the interior of the pancreatic duct, The interior means these glands all will communicate with what? Pancreas, liver. They all will be communicating with the gastrointestinal tract only, no? Therefore, the epithelium lining of the gastrointestinal tract will be lining the bile duct also and the bile canaliculi also. Therefore, they are all derived from endoderm. They are all derived from endoderm. The secretory glands of the inner mucous membrane of the GI tract, that all is derived from endoderm. Next, you remember regarding that skin, epidermis part of the skin is derived from the ectoderm, whereas the dermis part of the skin is derived from mesoderm. Uh, and the appendages of the skin, they all will be derived from the ectoderm only. ಈ ಸ್ಕಿನ್ ನೀವು ಎಕ್ಟೋಡಮ್ ಅಂತ ಡಿರೈವ್ ಆಗ್ತದೆ ಅಂತ ಹೇಳಿ ಗೊತ್ತಿದ್ರೆ ಎಪಿಡಮಿಸ್ ಪಾರ್ಟ್ ಆಫ್ ದ ಸ್ಕಿನ್, ಆವಾಗ ಅಪೆಂಡೇಜಸ್ ಆಫ್ ದ ಸ್ಕಿನ್, ಹೇರ್, ನೇಲ್ಸ್, ಮತ್ತೆ ಹೇರ್, ನೇಲ್ಸ್, ಸ್ವೆಟ್ ಗ್ಲ್ಯಾಂಡ್, ಸಿಬೇಷಿಯಸ್ ಗ್ಲ್ಯಾಂಡ್, ಇದೆಲ್ಲ ಎಕ್ಟೋಡಮ್ ಇಂದ ಡಿರೈವ್ ಆಗ್ತದೆ ಅಂತ ಗೊತ್ತಾಗ್ತದೆ. ಮತ್ತೆ ಕಂಪ್ಲೀಟ್ ನರ್ವಸ್ ಸಿಸ್ಟಮ್, ಒಂದೊಂದು ಸಿಸ್ಟಮ್ ಇಟ್ಕೊಳ್ಳಿ. ಫಸ್ಟ್ಗೆ ನರ್ವಸ್ ಸಿಸ್ಟಮ್ ಇಸ್ ಡಿರೈವ್ ಫ್ರಮ್ ಎಕ್ಟೋಡಮ್. Nervous system is derived from ectoderm. Okay. Mesoderm, cardiovascular system, muscular system, locomotor system, ಎಲ್ಲ ಇದೆಲ್ಲ mesoderm ಇಂದ. ಮಸಲ್ಸ್ ಎಲ್ಲ, ಈವನ್ ಸ್ಮೂತ್ ಮಸಲ್ಸ್ ಆಫ್ ದ ಗ್ಯಾಸ್ಟ್ರೋಇಂಟೆಸ್ಟೈನಲ್ ಟ್ರ್ಯಾಕ್ಟ್ ಎಲ್ಲ. ಎಂಡೋಡರ್ಮ್ ಒಳಗಿನ ಭಾಗ ಎಂಡೋಥೆಲಿಯಲ್ ಲೈನಿಂಗ್ ಆಫ್ ದ GI tract, endothelial lining of the pancreatic duct, endothelial lining of the bile duct, the epithelial lining of the urinary bladder, except trigon. ಇದು ಒಳಗಿನ ಭಾಗ ಎಲ್ಲ ಇದೆಯಲ್ಲ, that is derived from endoderm. ಹಾಗೆ ನೆನ್ಪಿಟ್ಕೊಳ್ಲಿಕ್ಕೆ ಆಗ್ತದೆ. ಅರ್ಥ ಆಯ್ತಾ? ಓಕೆ, ನೆಕ್ಸ್ಟ್
Thank you
ಕ್ವೆಶ್ಚನ್.
so much. Thank you so much for the explanation, sir. The next question is: does we correlate your answer with Ayurvedic topic Garbha Sharira?
No. And see here, the questions will be asked in such a way that if they ask for month-wise development of fetus, this is the part where we can correlate. But the question will be like modern question will be that month-wise development of, they will not ask Masanumasika Garbhavriddhi. If they ask Masanumasika Garbhavriddhi, better to write Masanumasika Garbhavriddhi, either the question only. If the modern question is asked month-wise development of fetus, better to write what is there in my PPT. Month-wise fetal development regarding first month, second month, third month, fourth month, fifth month. It is there in Tortora. From Tortora only I copied it. You can take it from Tortora. That is available regarding the month-wise development is there. ಬಟ್ ಯಾಕಂತ ಹೇಳಿದ್ರೆ, ನಾನು ಯಾಕೆ ಹೇಳೋದು ಅಂತ ಹೇಳಿದ್ರೆ, ಇಲ್ಲಿ ಕರೆಕ್ಷನ್ ಮಾಡುವಾಗ ಇದು ಮಾಡರ್ನ್ ಟಾಪಿಕ್ ಬಂದದ್ದು, ಇದಕ್ಕೆ ಆಯುರ್ವೇದದ್ದು ಬರ್ದಿದ್ದಾರೆ, ಇದಕ್ಕೆ ನಾನು ಮಾರ್ಕ್ಸ್ ಕೊಡುವುದಿಲ್ಲ ಅಂತ ಹೇಳಿ ಕೆಲವರು ವ್ಯಾಲ್ಯೂಷನ್ ಮಾಡ್ತಾರೆ. ಆ ಥರ ಆಗ್ಬಾರ್ದಲ್ಲ. ನೀವು ಮಾರ್ಕ್ಸ್ ಲೂಸ್ ಮಾಡಬಾರ್ದಲ್ಲ, ಲಾಸ್ ಆಗ್ಬಾರ್ದಲ್ಲ. ಅದಕ್ಕೆ ಮಾಡರ್ನ್ ಕ್ವಶನ್ ಕೇಳಿದ್ರೆ ಮಾಡರ್ನ್ ನಲ್ಲೇ ಉತ್ತರ ಕೊಡಿ. ಆಯುರ್ವೇದದಲ್ಲಿ ಆಯುರ್ವೇದ ಅನುಸಾರವಾಗಿ ಕೇಳಿದ್ರೆ ಆಯುರ್ವೇದ ಅನುಸಾರವಾಗಿ ಉತ್ತರ ಕೊಡಿ. ಈಗ ಸ್ಪರ್ಮೆಟೋಜೆನೆಸಿಸ್ ಅಂತ ಕೇಳಿದ್ರು, ನೀವು ಶುಕ್ರದ ಬಗ್ಗೆ ಬರೆದ್ರಿ, ಸರಿ ಆಗ್ತದ? ಆಗುದಿಲ್ಲ. ಊಜೆನೆಸಿಸ್ ಕೇಳಿದ್ರು, ಆರ್ತವ ಬಗ್ಗೆ ಬರೆದ್ರಿ, ಆಗುದಿಲ್ಲ. ಅದು ಸ್ಪರ್ಮೆಟೋಜೆನೆಸಿಸ್ ಬಗ್ಗೆ ಬರೀಬೇಕು 5 ಮಾರ್ಕ್ ಕೇಳುವಾಗ. ಈಗ 10 ಮಾರ್ಕಿನ ಕ್ವಶನ್ ಈಗ ಸ್ಪರ್ಮೆಟೋಜೆನೆಸಿಸ್ ಕೇಳಿದ್ರು ಅಂತ ಹೇಳಿದ್ರೆ ನೀವು ಶುಕ್ರದ ಬಗ್ಗೆ ಬರೀರಿ, ತೊಂದರೆ ಇಲ್ಲ, ಸ್ವಲ್ಪ ಆಡ್ ಮಾಡಿ. 10 ಮಾರ್ಕಿಗೆ ಕೇಳಿದ್ರು ಅಂತ ಹೇಳಿದ್ರೆ, ಆದ್ರೆ 5 ಮಾರ್ಕಿಗೆ ಸ್ಪರ್ಮೆಟೋಜೆನೆಸಿಸ್ ಕೇಳಿದ್ರೆ ಸ್ಪರ್ಮೆಟೋಜೆನೆಸಿಸ್ ಬರೀಬೇಕು.
Okay, sir. Thank you, sir. Thank you for the beautiful explanation. There's another most frequently asked question in our whole lecture series, sir. That is, how many diagrams we should write for 5 marks? How many diagrams we should write for 10 marks? And for which questions diagrams are really important? Should we draw a big one or small one? O- What exactly we should decide while drawing diagrams? This was the most frequently asked question, sir. And there was also another question, and that student asked whether we could use color pencils for doing diagrams. So this
No,
was the
no, no.
most frequent question, sir.
Color pencils are not allowed actually. Black pen and pencil. One pencil, first you trace in a pencil, and over that you write it in a black pen. Why? Because the answer booklets will be scanned and they will be uploaded. And for digital valuation, that diagrams, if we should see properly, ಕಾಣ್ಬೇಕು ಅಂತ ಇದ್ರೆ you should mark it in a dark pen above the ಅಥವಾ ಡಾರ್ಕ್ ಪೆನ್ಸಿಲ್ ಯೂಸ್ ಮಾಡಬೇಕು. ತುಂಬಾ ಅದು ಸ್ಕ್ಯಾನ್ ಮಾಡುವಾಗ ಕರೆಕ್ಟಾಗಿ ಕಾಣಬೇಕು. ಸ್ಕ್ಯಾನ್ ಮಾಡುವಾಗ ಕರೆಕ್ಟಾಗಿ ಕಾಣಬೇಕು. ಮತ್ತೆ ಎಷ್ಟು ಡಯಾಗ್ರಾಮ್ಸ್ ಬಿಡಿಸ್ಬೇಕು? ಈಗ ನೋಡಿ, ಡಯಾಗ್ರಾಮ್ಸ್ ನಲ್ಲಿ ನೀವು ಶೇಡಿಂಗ್ ಎಲ್ಲ ಮಾಡುವ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಅದನ್ನು ಪಾಶ್ ಮಾಡಿ ಆರ್ಟಿಸ್ಟಿನ ತರ ಶೇಡ್ ಎಲ್ಲ ಮಾಡುವ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಜಸ್ಟ್ ಎ ಸ್ಕೆಮ್ಯಾಟಿಕ್ ಡಯಾಗ್ರಾಮ್ ವಿತ್ ಲೇಬಲಿಂಗ್ ಅಷ್ಟು ಮಾಡಿದ್ರೆ ಸಾಕು. ಅದು ಅರ್ಥ ಆಗ್ಬೇಕು. ಆ ಡಯಾಗ್ರಾಮ್ ನೋಡಿದ ಕೂಡಲೇ ನಿಮಗೆ ವಿಷಯ ಗೊತ್ತುಂಟು. ಫಾರ್ಮೇಶನ್ ಆಫ್ 3 germ layers, ಅದರಲ್ಲಿ ಫಾರ್ಮೇಶನ್ ಆಫ್ ಹೈಪೋಬ್ಲಾಸ್ಟ್, ಎಪಿಬ್ಲಾಸ್ಟ್, ಫಾರ್ಮೇಶನ್ ಆಫ್ ದ ಯಾಕ್ ಸ್ಯಾಕ್, ಆಮೇಲೆ ಫಾರ್ಮೇಶನ್ ಆಫ್ ದ ಪ್ರಿಮಿಟಿವ್ ಸ್ಟೇಟ್, ಫಾರ್ಮೇಶನ್ ಆಫ್ ಪ್ರೋಕಾರ್ಡಲ್ ಪ್ಲೇಟ್. ಈ ಡಯಾಗ್ರಾಮ್ಸ್ ಎಲ್ಲ ಬಿಡಿಸ್ಬೇಕಾಗ್ತದೆ. ಈ ಡಯಾಗ್ರಾಮ್ ಎಲ್ಲ ಬಿಡಿಸದೆ ಫಾರ್ಮೇಶನ್ ಆಫ್ 3 germ layers ನಿಮ್ಮ ಆನ್ಸರ್ ಅಷ್ಟು ಸ್ಯಾಟಿಸ್ಫ್ಯಾಕ್ಟರಿ ಇರುವುದಿಲ್ಲ ಡಯಾಗ್ರಾಮ್ಸ್ ಬಿಡಿಸದಿದ್ದರೆ. ಆವಾಗ ಏನು ಮಾಡ್ಬೇಕಲ್ಲಿ? ಸ್ಕೆಮ್ಯಾಟಿಕ್ ಡಯಾಗ್ರಾಮ್ ಬಿಡಿಸ್ಬೇಕು. ನೀವು ಶೇಡಿಂಗ್ ಎಲ್ಲ ಮಾಡೋದು ಬೇಡ. ನಾನು ಹೇಳುದು ಸ್ಕೆಮ್ಯಾಟಿಕ್ ಡಯಾಗ್ರಾಮ್ ಬಿಡಿಸಿ ಜಸ್ಟ್ ಲೇಬಲ್ ಮಾಡಿ ಹೈಪೋಬ್ಲಾಸ್ಟ್, ಎಪಿಬ್ಲಾಸ್ಟ್, the ectoderm, endoderm and mesoderm, primitive state, procardium. They form that the primitive state proliferation of the cells, the interembryonic mesoderm is formed. ಅದನ್ನ ಇದು ಮಾಡಿದ್ರೆ ಆಯ್ತು. ಅಂದ್ರೆ ಡಯಾಗ್ರಾಮ್ ಈಗ ಕೆಲವು ಇಂಪಾರ್ಟೆಂಟ್ ಟಾಪಿಕ್ 10 ಕ್ವಶನ್ಗೆಲ್ಲ ಜಾಸ್ತಿ ಬೇಕಾಗ್ತದೆ. 5 ಮಾರ್ಕ್ಸ್ ಕ್ವಶನ್ಗೆಲ್ಲ 1 ಅಥವಾ 2 ಇದ್ರೆ ಸಾಕು. ಆ 1 ಸಾಕು ಅಥವಾ 2 ಸಾಕು. ಆದರೆ ಅದನ್ನು ನೀವು ಡಾರ್ಕ್ ಪೆನ್ಸಿಲ್ ಅಥವಾ ಪೆನ್ನಿನಲ್ಲಿ ಇದು ಮಾಡಬೇಕು. ಯಾಕಂದ್ರೆ ಶೇಡ್ ಮಾಡಿದ್ದು ನಮಗೆ ಕಾಣಬೇಕು ವ್ಯಾಲ್ಯೂಯೇಷನ್ ಮಾಡುವಾಗ. ನೀವು ಡಾರ್ಕ್ ಪೆನ್ಸಿಲ್ ಯೂಸ್ ಮಾಡದಿದ್ರೆ ಅದು ಲೈಟ್ ಆಗ್ತದೆ. ಸ್ಕ್ಯಾನಿಂಗ್ ಮಾಡುವಾಗ ಲೈಟ್ ಆಗ್ತದೆ. ಆದ ಕಾರಣ ಅಂಡ್ ಡೋಂಟ್ ಯೂಸ್ ದ ಕಲರ್ ಪೆನ್ಸಿಲ್ಸ್. ಡೋಂಟ್ ಯೂಸ್ ದ ಕಲರ್ ಪೆನ್ಸಿಲ್ಸ್. ಕಲರ್ ಪೆನ್ಸಿಲ್ಸ್ ಎಲ್ಲ ಬೇಕಂತಿಲ್ಲ. ರೆಡ್ ಕಲರ್ ತೋರಿಸ್ಬೇಕು, ಗ್ರೇನ್ ಬ್ಲೂ ಕಲರ್ ತೋರಿಸ್ಬೇಕು, ಏನು ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ದಟ್ ಇಸ್ ನಾಟ್ ಫಾರ್ ದ ಎಕ್ಸಾಮ್.
Thank you,
ಮತ್ತೆ
sir. Continuing with the same question, another student is asking if they ask Ayurveda topics to explain, should we dog diagrams from modern aspect and label?
No. Ayurveda portion, you write it Ayurvedic portion only. Not necessarily. ಈಗ ಶುಕ್ರ ಕೇಳಿದ್ರು, ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದ ಸ್ಪರ್ಮ್ ಬರೆಯುವ ಅವಶ್ಯಕತೆ, ಡಯಾಗ್ರಾಮ್ ಬಿಡಿಸುವ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಇಲ್ಲ. ನೀವು ಸ್ಫಟಿಕಾಭಂ ದ್ರವಂಸ್ಥಿದ್ದಂ ಮಧುರ ಮಧುಬಂಧಿಚ ಶುಕ್ರಮಿಚ್ಛತಿ ಕೇಚಿತು ಕೈಲಶಬ್ದ ನಿಬಂತದ ಅಂತ ಹೇಳಿ ನೀವು ಆ ಶ್ಲೋಕಗಳನ್ನೆಲ್ಲ ಬರೆಯದಿದ್ದರೆ ನಿಮಗೆ ಆ ಮಾರ್ಕ್ಸ್ ಸಿಗುದಿಲ್ಲ. ಆದ ಕಾರಣ ಆಯುರ್ವೇದ ಪೋಷನಿಗೆ ಆಯುರ್ವೇದವನ್ನೇ ಬರೀರಿ. ಅಲ್ಲಿ ನೀವು ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದ ಸ್ಪರ್ಮ್ ಬಿಡಿಸುವ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಶುಕ್ರ ಕೇಳಿದ್ರೆ, ಆರ್ತವ ಕೇಳಿದ್ರೆ ಆ ಶಶಾಸು ಪ್ರತಿಮ ಮತ್ತು ಯದ್ವಾಲ ಅಕ್ಷರ ಸೋಪಂ ತದಾರ್ಥಂ ಪ್ರಶಂಸಂತಿ ಯದ್ವಾಸವು ನವಿರಂಜೆ ಅಂತ ಹೇಳಿ ಆರ್ತವದ ಬಗ್ಗೆ ನೀವು ಶ್ಲೋಕಗಳನ್ನು ಕರೆಕ್ಟ್ ಆಗಿ ಶುದ್ಧ ಶುಕ್ರ, ಶುದ್ಧ ಆರ್ತವ, ಅದರ ಬಗ್ಗೆ ವರ್ಣನೆ ಕೊಡ್ಬೇಕಷ್ಟೇ ಹೊರತು ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದ ಸೆಕೆಂಡರಿ ಊಸೈಟ್ ಅದು ಬೇಕಂತಿಲ್ಲ. ಬೇಡನೇ ಬೇಡ. ಆಯುರ್ವೇದಕ್ಕೆ ಡಯಾಗ್ರಾಮ್ಸ್ ಬೇಡ. Okay.
Thank you, sir. There's another question, sir. Sir, tell about time management in exam, like how we should allot the time for 10 marks, for 5 marks, and MCQs. How many minutes we should for each section?
ನಾನು ಯಾವಾಗಲೂ ಇಲ್ಲಿ ಪ್ರಯೋಗ ಮಾಡುವುದು ಏನಂತ ಹೇಳಿದ್ರೆ ಕಾಲೇಜ್ನಲ್ಲಿ 180 minutes divided by 100. Andre 180 minutes is there for you people, and divided by 100 marks means 1.8 minutes per 1 mark. Means for 5 marks, it is maximum is 8 to 9 minutes. Listen here. 8 to 9 minutes. 10 mark question ಗೆ ಅದರದ್ದು ಡಬಲ್ ಅಂದ್ರೆ 18 minutes. ಅದೇ 1 mark question ಗೆ ಓನ್ಲಿ 1 minute ಅಥವಾ 1.5 ನಿಮಿಷ ಸಾಕು. ಆ, ನೀವು ನೀವು ಇದು ಈ ತರ ಪ್ಲಾನ್ ಮಾಡಬೇಕು ಫಸ್ಟಿಗೆ. ಅಂತ ಹೇಳಿದ್ರೆ ಈಗ ನಿಮಗೆ ತುಂಬಾ ಗೊತ್ತುಂಟಂತ ವಿಷಯ, ಒಂದು ಆನ್ಸರ್ ನಲ್ಲಿ ತುಂಬಾ ವಿಷಯ ಗೊತ್ತುಂಟು, ಇನ್ನೊಂದು ಆನ್ಸರ್ ನಲ್ಲಿ ಸ್ವಲ್ಪ ವಿಷಯ ಮಾತ್ರ ಗೊತ್ತಿರೋದು ಅಂತಿದ್ರೆ, ಫಸ್ಟಿಗೆ ಆ ಆನ್ಸರ್ ಅನ್ನು ಬರೆದು ಸ್ವಲ್ಪ ಸ್ಪೇಸ್ ಬಿಡಿ. ಅಲ್ಲಿ ನಿಮ್ಮ 9 ನಿಮಿಷ ಆದ ಕೂಡಲೇ ಒಂದು ಸ್ವಲ್ಪ ಸ್ಪೇಸ್ ಬಿಡಿ, ಬಿಟ್ಟು ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ಆನ್ಸರ್ಗೆ ಹೋಗಿ. ನೆಕ್ಸ್ಟ್ ಆನ್ಸರ್ ಅನ್ನು ಅಟೆಂಡ್ ಮಾಡಿ. ಅಟೆಂಡಿಂಗ್ ಆಲ್ ದ ಕ್ವಶನ್ಸ್ ಇಸ್ ಇಂಪಾರ್ಟೆಂಟ್. Writing one question in detail and another question not at all attempting, they will not give 6 out of 5. They will give maximum 4 to 4.5 out of 5. Therefore, you divide the 180 minutes by 100 marks, 1.8 minutes per 1 mark, and total 9 marks will be there for 5 marks question, 18 marks for 10 mark question.
ಥ್ಯಾಂಕ್ ಯು ಸರ್. ಸರ್, ಸ್ವಲ್ಪ ಜನಕ್ಕೆ ಈಗ ಎಕ್ಸಾಮ್, ಈಗ ಕೆಲವೊಬ್ರು ಪ್ರಿಪ್ರೇಷನ್ ತುಂಬಾ ಲೇಟ್ ಆಗಿ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ದಾರೆ ಸರ್. ನಾವು ಆಲ್ಮೋಸ್ಟ್ ಲೆಸ್ ದನ್ ಅ ಮಂತ್ ಇಸ್ ದೇರ್. ಸೊ ಅವ್ರು ಕೆಲವೊಬ್ರು ಅಂದ್ರೆ ಯಾವ ರೀತಿ ಅಂದ್ರೆ ಭಯ ಆಗ್ತಿದೆ, ಲೈಕ್ ಹೌ ವಿ ಕುಡ್ ಫೇಸ್ ದಿಸ್ ಇನ್ ಸಚ್ ಲೈಕ್ ದೇ ಸ್ಟಾರ್ಟೆಡ್ ಪ್ರಿಪೇರಿಂಗ್ ನೌ ಸರ್. ಸೊ ಅವ್ರಷ್ಟೇ ಅಲ್ಲ ಸರ್, ಸುಮಾರು ಜನ ಈಗ ಪ್ಯಾಟರ್ನ್ ಚೇಂಜ್ ಇರೋದ್ರಿಂದ, ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ನಮಗೆ 10 marks 2 ಕ್ವಶನ್ಸ್ ಇತ್ತು, ಈಗ ಇವ್ರಿಗೆ 10 marks 4 ಕ್ವಶನ್ಸ್ ಇದೆ. ಸೊ ಅಂಡ್ ಆಲ್ಸೋ ಪೇಜಸ್ ಇನ್ಕ್ರೀಸ್ ಮಾಡಿದ್ದಾರೆ, ಫ್ರಮ್ 5 to 6 pages ಮಾಡಿದ್ದಾರೆ. ಸೊ ದೇ ಆರ್ ಫಿಯರಿಂಗ್ ಆಫ್ ಎಕ್ಸಾಮ್ಸ್. ಸೊ ಅವ್ರಿಗೆ ಏನ್ ಅಡ್ವೈಸ್ ಕೊಡ್ಬೋದು ಸರ್?
See, now also time is more in your hand. You can, if you study seriously from now onwards also, I will tell that you will pass in the exam. You can ಮನಸ್ಸು ಕೊಟ್ಟು ಈ ಶ್ಲೋಕಗಳನ್ನೆಲ್ಲ ಬ್ಯಾಟ್ ಮಾಡ್ಲಿಕ್ಕೆ ಈಗ ಸ್ವಲ್ಪ ಕಷ್ಟ ಆಗ್ಬೋದು. ಆದರೆ ಶ್ಲೋಕಗಳನ್ನೆಲ್ಲ ಮೊದಲೇ ಬ್ಯಾಟ್ ಮಾಡಿ ಇಟ್ಟಿದ್ದಾರೆ, ಅದನ್ನು ರಿವೈಸ್ ಮಾಡಿ. ಮತ್ತೆ ಬಾಕಿದೆಲ್ಲ ಓದಿಕೊಳ್ಳಿ. ಆಯುರ್ವೇದ ಪೋಷನ್ ಬರೆಯುವಾಗ ಶ್ಲೋಕಗಳು ಬೇಕಾಗ್ತದೆ. ಮತ್ತೆ ಎಲ್ಲ ಮಾಡರ್ನ್. ನೀವು ಈಗಲೇ ಓದಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ರು ಕೂಡ ಮನಸ್ಸು ಕೊಟ್ಟು ಓದಿ, ಭಯ ಪಡಬೇಡಿ. ಎಕ್ಸಾಮ್ ಅನ್ನು ಫೇಸ್ ಮಾಡ್ಲಿಕ್ಕೆ ಧೈರ್ಯ ಮಾಡ್ಕೊಳ್ಳಿ. ಓಲ್ಡ್ ಕ್ವಶನ್ ಪೇಪರ್ಗಳನ್ನು ಸಾಲ್ವ್ ಮಾಡಿ. ಟೈಮ್ ವೇಸ್ಟ್ ಮಾಡಬೇಡಿ, ಇನ್ನು ಮುಂದೆ ಟೈಮ್ ವೇಸ್ಟ್ ಮಾಡಬೇಡಿ. ನಿಮ್ಮ ಕೈಲಿ ಇದೆ, ಇನ್ನು ಕೂಡ ಜುಲೈ 25 ಇನ್ನು ಕೂಡ ಬರ್ಬೇಕಷ್ಟೇ. ಚೆನ್ನಾಗಿ ಓದಿ ಪಾಸ್ ಆಗ್ಬೋದು. ಮಸ್ಟ್ ನೋಗೆ ಮೋರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಕೊಡಿ. ಡಿಸೈರಬಲ್ ಟು ನೋ, ನೈಸ್ ಟು ನೋ ಲಾಸ್ಟಿಗಿಡಿ. ಆಯ್ತಾ? ಮಸ್ಟ್ ನೋಗೆ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಹೆಚ್ಚಿಗೆ ಕೊಡಿ. ಡಿಸೈರಬಲ್ ಟು ನೋ, ನೈಸ್ ಟು ನೋಗೆ ಮತ್ತೆ ಇಂಪಾರ್ಟೆನ್ಸ್ ಕೊಡಿ. ಅದಕ್ಕೆ ನಾನು ಹೇಳಿದಲ್ಲ, ಸೆಕ್ಸ್ ಡಿಟರ್ಮಿನೇಷನ್ ಅಂಡ್ ಸೆಕ್ಸ್ ಡಿಫ್ರೆನ್ಸಿಯೇಷನ್ಗೆ ಹೆಚ್ಚಿಗೆ ಇಂಪಾರ್ಟೆನ್ಸ್ ಕೊಡುವ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಆ ಕ್ವಶನ್ ಯಾರಿಗೆ ಒಳ್ಳೆ ಸ್ಕೋರ್ ಮಾಡಬೇಕೋ ಅವರು ಅದನ್ನು ನೋಡ್ಕೊಳ್ಳಿ. ಯಾರಿಗೆ ಪಾಸ್ ಆಗಬೇಕು ಅಂತ ಇದ್ರೆ ಮಸ್ಟ್ ನೋವನ್ನು ಕಂಪ್ಲೀಟ್ ಮಾಡಲೇಬೇಕು. ಓಕೆ,
Thank you, sir. There's another question. Next question is from a student called Spurti. She is asking, does questions repeat from old question papers?
Yes, old question papers are very important. Usually, ಎಕ್ಸಾಮ್ ಇದಕ್ಕೆ ಕ್ವಶನ್ ಪೇಪರ್ ಸೆಟ್ ಮಾಡುವಾಗಲೂ ಕೂಡ ಓಲ್ಡ್ ಕ್ವಶನ್ ಪೇಪರ್ಗಳನ್ನು ನೋಡಿ ಕೆಲವರು ಸೆಟ್ ಮಾಡುವ ಅಭ್ಯಾಸ ಇಟ್ಕೊಂಡಿರ್ತಾರೆ. ಆದ ಕಾರಣ ಓಲ್ಡ್ ಕ್ವಶನ್ ಪೇಪರ್ ಇಸ್ ಆಲ್ಸೋ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. Important. Next question.
Thank you, sir.
Important. Next question.
Thank you, sir. The next question is from Apsana. How to represent answers for 10 marks question? Like how to structure it, sir?
Say structure
Say structure.
ಕ್ವಶನ್ ನಲ್ಲಿ ಮೇನ್ ಪಾಯಿಂಟ್ಸ್ ಗಳನ್ನು ನಾನು ಕವರ್ ಮಾಡಿದ್ದೇನೆ ಇಲ್ವಾ ಅಂತ ನೋಡ್ಕೊಳ್ಬೇಕು. ಈಗ ನಾನು ಅದು ಪಿ ಪಿ ಟಿಯಲ್ಲಿ ತೋರಿಸಿದೆ. ಫಾರ್ಮೇಶನ್ ಆಫ್ ಪ್ಲಾಸೆಂಟಾದಲ್ಲಿ ಫಾರ್ಮೇಶನ್ ಆಫ್ ಪ್ರೈಮರಿ ವಿಲ್ಲೈ, ಸೆಕೆಂಡರಿ ವಿಲ್ಲೈ, ಟರ್ಷರಿ ವಿಲ್ಲೈ, formation of decidua, and formation which are the three layers which are responsible for the formation of placenta: trophoblast, extraembryonic mesoderm, as well as the decidual vessels. They contribute for the formation. ಸ್ಟೆಪ್ ವೈಸ್ ನಲ್ಲಿ ಕರೆಕ್ಟ್ ಆಗಿ ನೀವು ಪ್ರೆಸೆಂಟೇಷನ್ ಹೆಡ್ಡಿಂಗ್ ವಿತ್ ಸಬ್ ಹೆಡ್ಡಿಂಗ್ ಹಾಕಿ ಚೆನ್ನಾಗಿ ಪ್ರೆಸೆಂಟ್ ಮಾಡಿದರೆ ನಿಮಗೆ ಒಳ್ಳೆ ಮಾರ್ಕ್ಸ್ ಪಾಯಿಂಟ್ ಟು ಪಾಯಿಂಟ್. ಇದು ಇಟ್ ಈಸ್ ನಾಟ್ ನೆಸೆಸರಿ ಟು ಫಿಲ್ ದ ಪೇಜಸ್. It is necessary to include all the points, important points in the answer. ನಾವು ಪಾಯಿಂಟ್ಸ್ ಇದೆಯಾ ನೀವು ಕವರ್ ಮಾಡಿದ್ದೀರಾ? ನೀವು ಫಾರ್ಮೇಶನ್ ಆಫ್ ಪ್ರೈಮರಿ ವಿಲ್ಲ ಬರ್ದಿದ್ದೀರಾ? ಫಾರ್ಮೇಶನ್ ಆಫ್ ಸೆಕೆಂಡರಿ ವಿಲ್ಲ ಬರ್ದಿದ್ದೀರಾ? ಟರ್ಷರಿ ವಿಲ್ಲ ಎಲ್ಲ ಬರ್ದಿದ್ದೀರಾ? ಓ, ಈ ಆನ್ಸರ್ ಚೆನ್ನಾಗಿದೆ, 8/10 ಕೊಡುವ ಅಂತ ಹೇಳಿ ಅನಿಸ್ತದೆ. ಹಾಗೆ ಎಲ್ಲ ಪಾಯಿಂಟ್ಸ್ಗಳು ಕವರ್ ಮಾಡಿದ್ದೀರಾ ಅಂತ ನೋಡ್ಕೊಳ್ಬೇಕು. ಅದಕ್ಕೆ ನಾನು ಹೇಳಿದ್ದು, ನೀವು ಟೈಮ್ ಡಿವೈಡ್ ಮಾಡ್ಕೊಳ್ಳುವಾಗ 1.8 minutes ಫಾರ್ 1 mark ಅಂತ ಹೇಳಿದ್ರಲ್ಲ, ಅದನ್ನು 1.5 ತಗೊಳ್ಳಿ. 1.5 ಫಾರ್ 1 mark. ಉಳಿದದು 0.3 minutesಗಳನ್ನು ಲಾಸ್ಟಿಗೆ ನೀವು ಆನ್ಸರ್ ಅನ್ನು ಪೇಪರ್ ತಿರುಗಿಸಿ ನೋಡ್ಲಿಕ್ಕೆ ಯೂಸ್ ಮಾಡಿಕೊಳ್ಳಿ. ಎಲ್ಲ ಕವರ್ ಮಾಡಿದ್ದೇನೋ ಇಲ್ವಾ ಅಂತ ನೋಡ್ಕೊಳ್ಳಿಕ್ಕೆ ಯೂಸ್ ಮಾಡಿ. ಮತ್ತೆ
Thank you, sir. I think that's all from our question and answer session, sir. Uh, Really, thank you for agreeing for taking a session on embryology, sir. Actually, embryology is such a vast topic, and constraining it to the time constrictions that we asked for is really a challenging task. Explaining the whole topic in just an hour, but thanks for agreeing to do that, sir. On behalf of my whole team and the whole Jignasa organization, we are really grateful that you agreed to host an session for us, and it was really an educative session. Even for me, it's helpful because I'm in third year. We have PTS, so it was really an educative session for me also. Um, so really thanks from my whole team and the organization. The team is really working very hard from past 2 months to get these things on online, to get these things on stage, to reach for more students, to get for them to get benefit out of this, because there's no single channel on YouTube that does that have a series like a structured topic wise series for anatomy. It's scattered. We faced difficulties during our first year when we were preparing for the exams. We didn't get a proper channel where every video was there. Even after listening in classes, it's I think it's our nature to go for YouTube in during our last minute preparations and to get like last minute preparations done. So we planned that if we do topic wise lecture series, then it will be easy for students to get all that on one single platform. There will be a no efforts from there or time wasting from them for searching the topic wise videos on multiple platforms. So it was planned based on this aim, and we also did note series that we have written the whole notes of first year. The team has written from the past 2 months again. So we are trying our best to help the exam writing batch of 21-22. So and I really am glad that you also helped us and took a part in contributing for the students, sir. We are really grateful to you.
I thank the
Oh, I thank
Jignasa team
the Jignasa team.
for making such a facility for the students. I also thank them to provide me a opportunity to express my thoughts. Thank you. All the best. All the best to the first year exam going
Exam
students.
going students.
Thank you, sir, and thank you, ma'am, too. Thank you. Thank you, sir. We will be ending our session now. And thank you, guys, for joining. Join us again on the same time tomorrow for another session of higher anatomy. Thank you all for joining.
Thank you.
Thank